So in 9.4 here, what we are going to be talking about is temperature and microbial growth. So again, in chapter 9, we're talking about how microbes grow. And so in each of the sections, we're talking about different things that affect microbial growth. So when we're talking about temperature and microbial growth, we're seeing something similar to pH, where we have a minimum growth temperature. So minimum growth temperature. And that, of course, is the lowest temperature where this organism, whatever organism we're talking about, can grow. Where it can grow. And then we have our maximum growth temperature. Of course, the maximum temperature where it can grow. And then we have our optimum growth temperature. And this is where our growth rate is the highest. So then we have terms similar to what we saw in pH. We have terms that are associated with different temperatures in this case. So where we find most of our organisms, especially ones when we're talking about human health, they are called mesophiles or mesophiles. And so these are organisms with their optimal growth at temperatures that are about 20 degrees Celsius to 45 degrees Celsius. So mesophiles or mesophiles are going to be where we're going to see something like all of our pathogenic bacteria for humans, for example, because we are at about 37 degrees Celsius. That's our 98.6 degree Fahrenheit. So we're at about 37 degrees Celsius, which means that we fit right in here. And so the organisms that live in us and on us that help us are mesophiles. And then those that are pathogenic to humans oftentimes are also mesophiles because they are at that optimum pH for them, or optimum temperature for them. The next group are the Psycrow tropes. So psychrotrophs are also called psychrotolerant. So psychrotroph and psychrotolerant are the same thing. So psychrotroph or psychrotolerant, their optimal growth temperature is going to be about 4 degrees Celsius to about 25 degrees Celsius. And so 25 to 30 degrees Celsius or 25 degrees Celsius really is about room temperature because room temperature is somewhere around in the mid 70s. So, and 25 degrees Celsius is around 77 or so. So room temperature oftentimes I think is a little bit lower than that, around 73, 74-ish. In houses, perhaps is a little bit higher, more in the middle 70s. So it's about room temperature. So psychrotrophs are psychrotolerant. So troph, remember, is how it obtains its nutrition. So nutritionally, it is something that likes a little bit colder environments, or tolerant or tolerates a little bit colder environments. Then we have the psychrophiles. And the psychrophiles are those that have their optimal growth temperatures at less than 0 degrees Celsius, up to about 20 degrees Celsius, whereas the optimal is at about 15 degrees Celsius for optimal growth. So our psychrophiles, philes meaning loving, so these are those that are psychrophiles are cold loving versus psychrotrophs or psychrotolerant are cold tolerant. So we see the lower temperature when it's psychrophiles. Then if we go the opposite direction, so this is where we're going colder, of course, than a mesophile. If we go warmer than a mesophile, then we have our thermophile. And our thermophiles have their optimal growth at temperatures that are between 50 degrees Celsius and 80 degrees Celsius. So quite warm. So thermophile, of course, phile meaning loving, and thermo meaning warm loving. But then we have one more above that. Those are the hyperthermophiles. And so hyperthermophiles are even higher than that. So their optimum growth temperature is at about 80 degrees Celsius, where we left off with the thermophiles, up to about 110 degrees Celsius. And this actually can go higher. So it could really be greater than 100, I guess if we said it the correct way, it would be greater than 80 degrees Celsius. So greater than 80 degrees Celsius. And it really depends where in that it goes. We've seen things that go all the way up to 120 degrees Celsius that live most optimally at that temperature. But then when we're looking at something like the heat vents at the bottom of the ocean, those can be up to 400 degrees Celsius. And there are still microbes that are living in that. So hyperthermophiles really have a great range. But what that means is anything really above 80 degrees Celsius, and we're really talking about 80 to 110 degrees Celsius. And then we have some outliers that are going to be much higher than that. So why is it that we see most organisms are going to be hanging out in the mesophile range, particularly those that affect humans, versus something colder or something warmer? And that's because we are going to look at that's because of the different effects that we see with low temperature and with high temperature on the actual organism. So let's talk about some of the low temperature effects here. So one, with a low temperature, we can lose membrane fluidity. So if we think of our plasma membrane and how it's this kind of mosaic of moving parts and moving pieces, there has to be a fluidity to it. So we have our unsaturated fatty acids that kind of become bumps in the road or little rocks on the surface, that our saturated fatty acids can kind of move around, and everything becomes and everything's pretty fluid. Now, if we take a mesophile, for example, and we put it into a cold environment, it's going to lose that membrane fluidity, right? Things get slower, things move slower in the cold. So the plasma membrane is not going to function as it needs to. Also, we can have damage due to ice crystals. So if we put an organism in something in quite cold temperatures, the actual water inside of the organism is going to start to freeze. And when it freezes, it forms crystals, and those crystals are sharp, and then they can burst the cell. They can break the cell wall, break the plasma membrane, or even before it gets to that point, it can just damage molecules within the organism. In general, reactions and diffusion is going to slow. So just like we get much, much slower in cold, so do other organisms. If you think about if your hands get really, really cold, say it's wintertime, you're out in the snow and you've been making snowballs without gloves on or something, over time it hurts to move your fingers, or it can be difficult to move your fingers, for example. That's because everything slows down when things get cold. So reactions slow down. Diffusion also slows down. And if it slows down too much, then we're not going to have the metabolic processes that are necessary for life when it slows down too much. So then that can be contributing to the death of something that is a mesophile, for example, and you put it in, say, 0 degrees Celsius. Then also our proteins, which remember our proteins are very important because they are our enzymes, or majority are enzymes. Our proteins will become too rigid. So they'll become too rigid to actually do their function. So again, slowing down the enzymes, slowing down the proteins, they become too rigid to even be enzymes anymore, and they can also denature. So they may fall apart just because they're trying to move, they're not moving, they just slow down and they stop working. So some of the high temperature effects are going to be kind of the opposite of this. We already know that it's going to denature proteins. So even in cold temperatures where things become rigid and slow down, when we increase the temperature, then proteins actually become denatured because they're moving around so quickly that those bonds are just going to be broken between the amino acids, creating the quaternary structure, the tertiary structure, and then even getting down into breaking apart the individual amino acids when we get into an extreme high heat situation. It's also going to denature our nucleic acids. Again, because we have lots of hydrogen ions, for example, those hydrogen bonds can move very quickly and change very quickly. When we increase the temperature, we start to break apart our bonds between our atoms, between our elements. It can also then increase membrane fluidity. So if we are increasing membrane fluidity, we can be increasing it so much that we actually hinder the processes that are important to that membrane. So if there isn't enough structure to actually go through the process, for example, of exocytosis or endocytosis, and it's so fluid that it can't form these structures, then we have a problem because we're not able to do these metabolic processes that are necessary. And of course, in extreme high temperatures, then all of these things in the membrane will start to fall apart, and then the cell will die anyway because of this very, very high increase in temperature. So looking at how these organisms that can live in low temperatures and can live in high temperatures is important. So we can take a look at their adaptations. So if we first take a look at adaptations of psychrophiles. So remember, psychrophiles are those that are in decreased temperatures. So if we're talking about decreased temperatures, we're talking about these items up here that we're trying to overcome through these adaptations. So for example, we see that proteins in psychrophiles are usually rich in hydrophobic residues. Hydrophobic. So if they are rich in hydrophobic residues, which means it's going to be sequestering itself more away from water, which would be freezing and forming those ice crystals, and kind of sequestering itself with other hydrophobic regions. And if we think about if you took olive oil, for example, and put it in the freezer, compared to if you take water and put it in the freezer, you know that it would take much longer, or not at all, for the olive oil to freeze in the freezer compared to water freezing in the freezer. So it's a much lower temperature with which we would have to, or that we would have to reach in order for olive oil, something that's hydrophobic, to freeze compared to water. So we have an increase in hydrophobic residues. We also see an increase in flexibility. And a lower number of secondary stabilizing bonds. So when we are talking about flexibility, we want to increase flexibility because if we are in lower temperatures, again, we decrease flexibility then. And we want to decrease the number of secondary stabilizing bonds in proteins, for example. So when we think about proteins, we have all of these, the alpha helices and the beta pleated sheets, and all of these form these secondary stabilizing bonds to hold the protein in a particular conformation. So in the case of an adaptation for cold weather, we're going to remove or decrease some of those secondary stabilizing bonds so that they are able to remain more fluid, so that things can be more fluid. It's not going to adjust the function of the protein, but we're just taking away some of those additional bonds so that it can be a little bit more fluid, so that when it is in lower temperatures, it is not going to become too solid to not be functional anymore. We also see antifreeze proteins in some of these organisms. So antifreeze proteins. And what this does is it decreases the freezing point of the cytoplasm. And if it decreases the freezing point of the cytoplasm, again, kind of back to that analogy of the oil versus water, if we decrease the freezing point, then an organism can live in much lower temperatures without freezing and dying. Also, they have lipids that are more made up of unsaturated fatty acids in order to remain more fluid. So lipids are unsaturated in order to remain fluid. So again, unsaturated fatty acids have those kind of kinks in the chains versus our saturated fatty acids, which are more straight. And these kinks are what are going to allow things to be more fluid. Rather than our saturated fatty acids, which can sit right next to each other, our unsaturated fatty acids have to have more space between them. So that maintains fluidity. And then growth rates are lower. So they go through the processes to grow much, much slower, and that's an adaptation. So we're not going to see 20-minute generation times with a Psycrow file, for example. It might be multiple hours or days or weeks. When we talk about our Psycrow files, remember we're talking about something that's very, very cold. So when we talk about freezing conditions and talk about those in regards to mesophiles, right, we're talking about them not being able to grow in freezing conditions or very, very cold conditions. However, we can do some things to allow mesophiles to continue existing in these freezing cold temperatures to then be used later. So, for example, we see this with pathogenic bacteria that we put in the refrigerator. So that's not freezing, but if we do put it in the freezer, sometimes when we thaw something out, it'll already have bacteria in it. It can grow when we thaw something out, especially if we thaw it out inappropriately, and then it can actually cause human disease. So even though we're talking about a pathogen that is a mesophile, it then was able to withstand those freezing temperatures for a period of time. And so we can have short-term storage of a mesophile in freezing conditions or refrigerated conditions. And then we can do this on purpose with very low temperatures. So if we talk about freezing, if conditions, if we have mesophiles, and some thermophiles, they can survive. And meaning that if we create conditions, some of these mesophiles and some of these thermophiles can survive. And we see this in laboratories for long-term storage. So if we want to continue cultivating an organism, but remember that when we're culturing an organism in the laboratory, the organism is going to be using up all the nutrients in the medium. And then once it uses up all the nutrients in the medium, it starts to die off. If it starts to die off, then we're going to be losing that organism, and it'll die completely over time if it has too many wastes built up and not enough nutrients available. If this is the case, then you're not going to be able to continue your research until you somehow get an initial stock again and start to grow it up again. So what scientists do in laboratories is they will actually go through the process of mixing this liquid culture with glycerol. And then what it does is that this glycerol is going to displace the water in the organism. As the glycerol displaces the water in the organism, glycerol is more of a hydrophobic molecule. So it's going to decrease the damage that's done by freezing the organism. So the culture will be placed in glycerol, and then it can be frozen. And it will be frozen at negative 80 degrees Celsius. So all the way down to negative 80 degrees Celsius for very long-term storage. So this is, again, so that in a laboratory you can make a huge amount of a particular culture. You can go in, you can add glycerol, and you can freeze all of that, keep it in cold long-term storage, and then all you need to do is bring it out, and then you need to warm it up again, and then you can start growing that organism again. It just kind of stays still. Also, we can take these cultures and we can freeze dry them, which is lyophilization. So lyophilization. And then we can store them as powders in sealed ampules. And this is oftentimes, I know for our laboratory at the college, but then also in laboratories across the country, we can order these organisms from a company, and what they've done is they do exactly this. They freeze dry the organisms, and then they send them to us in a container in these little ampules that are sealed, and then we need to rehydrate them. And we do that by adding a very particular amount of a solution that comes with the organism, and then incubate that for 24, 48 hours. And then from that point on, you can then culture it in the regular nutrient medium or any other medium, depending on the type of organism that you are culturing. Good. So now let's take a look at some of the adaptations of thermophiles. So adaptations of Thermo File. So what are these organisms going to do to withstand the high heat? So first example is the ratio of saturated fatty acids to polyunsaturated fatty acids is higher. So again, we're seeing the opposite that we saw before, and then this is to limit fluidity. Whoops. Fluidity. So again, we're going to have more saturated fatty acids in this case than polyunsaturated or just unsaturated fatty acids, because we want to try to make it less fluid, because it's going to be in an environment that of high heat, which is going to be making things move around more on its own. So in order to combat that, then we're going to have a higher ratio of saturated fatty acids to unsaturated. Another way that we can combat that is in the nucleic acids to have an increase in the guanine cytosine nucleic acids. Excuse me. So, and this is because our guanine cytosine, the way that I did this actually, was because there are 3 hydrogen bonds between the guanine and cytosine versus our adenine and thymine. There are only 2 hydrogen bonds. So we're actually going to see more GC combinations, because then it's going to make sure that those nucleic acids are more stable. So they've actually changed their genetic makeup in order to make it more stable at these higher temperatures. We also see that proteins have additional secondary ionic and covalent bonds. And they can even replace key amino acids to stabilize folding. So again, in this case, what we're doing is we're making more stabilized bonds, more ionic bonds, more covalent bonds. We replace amino acids to make things more stable, more folded, because we're trying to combat that increase in temperature, which makes things move a whole lot more. And so we want to decrease all of this movement. So when we think back for a moment here, most of our bacterial pathogens are going to be mesophiles. So they're going to be existing, most of them, between 20 degrees and 45 degrees Celsius. And so then sometimes things are going to be out of that area. And we're going to be taking a look at how our bodies are actually going to utilize things like increasing temperature to help our bodies fight off different organisms. But I want to say right now that it's not that our bodies increase temperature so much that it's going to take a mesophile and put it into a thermophile or hyperthermophile range. Our bodies don't get hot enough to kill the organism by temperature. If it did, then it would get so hot that it would kill our own cells by temperature. And sometimes that does happen if we have an extreme situation with an extreme high temperature, an extreme high fever, we can start damaging our own cells. And that's when someone would need to go to a hospital and get an ice water bath and things like that. That's an extreme circumstance when the body is not self-regulating. Our typical fever, when we as humans have a pathogen that we're trying to get rid of, if our fever goes up to about 104-ish, and even stays at 104 for multiple days, that's fine. And that's actually a good thing, because our body's doing what it's supposed to do. It's supposed to increase temperature, because what that actually does is it increases all of these metabolic processes. So just like we just spoke about, mesophiles like to be in this particular range. If we go up to the higher end of that range, then our cells actually work better. We go through more metabolic processes, which means we're making more ATP, which means we're more active, which means our immune system is more active, which means we have a better chance of fighting off bacteria. And so the temperature itself in a fever is beneficial for killing off bacteria, because we're making our body more efficient at getting the bacteria. And so that's why high temperatures are good. That's why high fever is good within parameters. So if we get above that 104 degree mark, Fahrenheit, of course, if we get above that 104 degree mark for a long period of time, that's when it would be important. Or if we get higher than that, if it continues to climb, then that's when we would want to seek medical care. because that's getting to where we are starting to do things like denaturing our proteins and affecting our nucleic acids and things like that, which is damaging to our own body. And then we would need some medical intervention at that point. So we will talk more about that, however, when we get to the end of the semester in Chapter 18, when we're taking a look at our immune system and how all of these pieces that we're learning throughout the semester about different types of bacteria, different pathogens, all kind of culminates at the end with how do our bodies deal with these different things now that we've learned about them. So you should be familiar with all of these terms, these minimum growth temperature, maximum growth temperature, optimum growth temperature, and then these different terms and the temperatures that they work best in. They're optimal growth temperatures for mesophiles or mesophiles, psychrotrophs, psychrophiles, thermophiles, and hyperthermophiles, and the adaptations. So why it is that a low temperature is bad, why it is that a high temperature is bad for organisms, and then how those organisms that are psychrophiles, for example, or thermophiles or hyperthermophiles are able to adapt to get beyond that and not die because of it.